Today I'll talk about species richness. Species richness is the number of different species represented in an ecological community, landscape, or region. As I told you earlier, that richness is simply a count of different species and it doesn't take into account of the species abundance or their relative abundance. Species richness can be estimated using different quantitative indices, and these indices have been developed by different wildlife biologists or conservationists named as Jackknife Richness Index, Margoliouth's Richness Index, or Macnin Richness Index. All these indexes have different expressions and calculate richness estimates. Jackknife Richness Index, we have expression which can be denoted by S bar is equal to S n minus 1 divided by n, close this expression in bracket, and here is k. So we have here S, n and k. S represents total number of species. Total number of species. N is number of quadrate sampled or number of plots sampled. Number of quadrate or plots sampled. K is unique species. Unique species. Number of unique species. Unique species are what? Are common species. These unique species are common species. In different samples, quadrates, or plots. Next index is Margoliouth index. It can be denoted by DMG is equal to S minus 1 divided by log. And clear this. Log n, and put this within bracket. So here we have S and n. S represents total number of species. Total number of species. And n, total number of individuals in the sample. Total number of individuals in sample. Next index is Macnin Richness Index. It can be denoted by D ml. It is equal to S divided by root n. So S represents total number of species. And n, it represents total number of individuals in a sample. Total number of individuals in a sample. Now we will see how to calculate species diversity. For species diversity, we have Shannon index, and often Shannon index have been represented in the form of Simpson index. Species diversity. It is the number of species in a community and their relative abundances. Species are not equally abundant. Some species occur in large percentage of samples, others are poorly represented. For example, some communities like a rainforest are much more diverse than others like desert. The expression for Shannon index can be denoted by H. Is equal to minus summation pi log in pi, or this expression can be written as summation bracket ni divided by n log now take log of this whole value ni divided by n. This pi, it represents here importance probability for each species. Importance probability each species. What is this probability and how we will calculate it? It will come from this value. It is ni divided by n. So we need the value of ni divided by n here. What this ni represent and what this n capital N represent here. Ni is importance value for each species. It is importance value for each species. And capital N represents total of importance value. Total of importance value. Simpson index. As often Shannon index or estimates are being represented by Simpson index, it can be denoted by D is equal to, it's a very small expression, 1 divided by summation pi ka square. It's very simple. And pi we know what is. We will get this value from the Shannon index value, and just have to make a square of that value, and it will provide a Simpson index. Next we have evenness index. This index can be represented by E is equal to H divided by, here we need value of log, so log S. H we know it's a Shannon value, and log S, what is S? S is number of species. Next index is similarity index. When we are interested to see similarity between different quadrids, plots or samples or study area, this index has been denoted by as, which is equal to 2C. This is also a very simplest index. A plus B, maybe plus C, and go on. So C, I have told you in the first slide expression that C are unique values, number of unique values, number of unique values. And A, B, C are study sites or study plots or quadrids, whatever. Number of species in study site 1, number of species in site 1. B is number of species in site 2 or etc. Here we will calculate the similarity. If we are interested in estimating dissimilarity between different study sites, so just we need to do one thing. Minus this value of similarity from 1 minus S, it will provide you value of dissimilarity. Right? We have one index, that is a dominance index. It is represented by C is equal to summation, and it is again a very smallest fraction, Ni divided by capital 1 ka whole square. Ni we know what it is. It is importance value for each species. Importance value for each species, and N is total of importance values. That's it. So we have discussed many indexes to estimate the species richness and species diversity. Tomorrow I'll discuss more properties of species diversity. Bye-bye.